Okay, our recording has started. Well, good afternoon, everybody. Thank you very much for taking the time to join our webinar roundtable discussion here from Biologics Consulting. My name is Chris Krahansel, and I have the privilege of overseeing all the business development and client engagement efforts here for our organization. And so, once again, just thank you for joining us today. Our topic that we have some of our experts here to discuss is new FDA CAR-T guidance. And so we're grateful that you came and that you tuned in. There will be some time at the end of today's discussion for some Q&A, and we strongly encourage you to reach out to today's presenters if you have any additional questions after our time is completed today. You can find them on LinkedIn, or you can go to our website, and they also have some contact information there that you're able to use to get in touch with them. So here at Biologics Consulting, we do what we do for the good of public health. And so without further delay, I want to introduce our moderator, David Pepperell. He is our non-clinical business unit. So David, take it away.
Thank you, Chris. Appreciate the ability to participate in this session. It's a very timely session on CAR-T therapies. We have two esteemed panelists today, members of my colleagues, Biologics Consulting. Our first panelist, Dr. Christopher Skull, joined Biologics in 2017 as a senior non-clinical consultant. Chris's experience spanned research, discovery, preclinical development, regulatory affairs, product development strategy, design and management of farm tox studies, writing and reviewing of FDA submissions, as well as GLP compliance oversight. Prior to joining Biologics, Chris served as Global Director of Discovery Sciences at Innovamune Biotherapeutics and served in multiple roles at Memorial Sloan Kettering Cancer Center in New York City. Our second panelist, Robert Kuttner, joined Biologics Consulting in May of 2022 and has over 22 years of cell and gene therapy experience. Robert has direct experience in discovery, process and analytical development, process characterization and validation, as well as authoring regulatory submissions. Robert also has experience in non-clinical and clinical assay development, and he supports process and analytical development for cell and vector products, comparability study design, as well as CMC regulatory filings. So Robert has extensive experience in industrial scale production for cell and gene therapies, and he is trained in GMPs and can advise on best practices for development of drug substances or drug products. So with that, I'd like to kick off the discussion relevant to this new 2024 FDA guidance document on CAR-T product development. So I'll start. This first question is for both panelists, but I'll start with Chris. Chris, to you, what stands out as most impactful in this new final FDA CAR-T guidance document?
Yeah, thanks, David. I think, it was eagerly anticipated, as we don't get a lot of cell therapy guidances since the last few years. On the non-clinical side, in particular for farm tox, I would say this one was a little bit light, and only 2 or 3 pages really going through some of the highlights for non-clinical. There was a few things that did stick out to me. First of all, they did reference for, more detailed farm tox guidance to go back to their 2013 guidance on cell and gene therapy. But in particular, in this 2024 guidance, I did notice that around the animal models was one issue there, where they're continuing to see favorably pharmacology studies with xenograft models, and rodents still seem to be acceptable for CAR-Ts, as well as an acceptance of using surrogate products where appropriate. We have a lot more complexity now for some of the newer CAR-T products, and being able to test those in a system with an intact immune system is also favorable. I also noticed sort of a second kind of point that was maybe not, called out specifically in its own subheading, but they did touch on issues around uncontrolled proliferation and really kind of use that wording as opposed to some of the wording they've used in the past where we've wondered, is it really necessary to do a tumorigenicity study? And so I think we're seeing a little bit of relaxing, even in the formal guidance around tumorigenicity. There is still plenty of comments there that there should be some assessment of proliferation, but not necessarily saying that it has to always be an in vivo tumorigenicity for CAR T products. And that's sort of lining up with what I've seen recently in the past few years with submissions going in for CAR Ts. The final thing that I also thought was interesting, the question we get from a lot of sponsors that are doing animal work for CAR T products is, how do I justify my dose that I want to use for the clinic? And again, although it wasn't a huge section of the guidance, I was interested to see that they did specifically state that rather than relying on some sort of golden calculation from your animal dose to human dose, that sponsors, particularly for CAR T products, can rely on previous clinical experience to guide dose selection. And I think that that's something a lot of sponsors should take into account, because there isn't always just a magic formula for translating your murine dose to humans. So those were a couple of things that stuck out for me, although again, it was a little light on farm tox for this particular guidance.
Yes, definitely. So thanks, Chris. Robert, what were your perceptions of this an updated document?
Yeah, thanks, David. I was on the same page as Chris. It wasn't too extensive in regards towards a lot of the updates. I think having been in the industry and seeing the trends, it is good to see them kind of get the guidances up to date with what has been the industry trends. But what stands out most to me, kind of looking through it, is the aspect of the how they further define the gene editing components that was not very clear in previous guidances, and ultimately the context towards the routine manufacturing aspects of some of these vectors and such that are being used. Because having been through a lot of product development, it's hard to create a blanket expectation in regards towards the release of these materials. And if some materials, such as the vectors, are only used at one time, a single time, it now has some guidance to kind of further support what are the expectations versus the routine productions, which you typically see, let's say, in an autologous cell therapy where multiple vector batches have to be generated. They, can further allow for appropriate specifications that would work in the latter setting versus, the more infrequent use or even one-time use to that end for some of the likely allogeneic materials and such, where they're generating a cell bank. And so I think overall that's very helpful for the industry, because again, kind of circling back to that aspect of, one size fits all for expectations on these. What also they gave some further definition in the active ingredients for the materials, kind of creates too much of an expectation for product development that might hinder products getting into the clinic, because of the amount that would be needed, is what would be needed for a larger scale production or a more routine one.
Oh, interesting. Yeah. I know certainly they're moving from, all the products have been generally autologous, but now we're seeing a lot of them move toward allogeneic products as well. So that, I think we'll touch on that in a moment or two. Chris, I know there's a lot of challenges with regard to developing these products and testing them in animals. So what do you see are some of the key limitations of some of the available animal models for testing CAR T product safety and efficacy?
Yeah, again, I think as some of the CAR T products become more complex, either targeting one or more CARs or one or more tumor antigens, or having different ways of expressing cytokines or interactions with other immune modulators, that it really becomes a challenge to find an animal model where all of the components of your product are biologically active. Even targeting the tumor antigen has been a challenge. If you use a xenograft model in immune-compromised mice, you can have a tumor tissue with the target, but that makes it challenging to really assess the potential for your CAR to bind that target that might be expressed in other tissues if it doesn't cross-react with mouse tissues. And, I think I would advise sponsors to consider cross-reactivity early rather than brushing it off and considering the fact that there might not be cross-reactivity, so you're just going to do a xenograft model. I think really in those early interactions with the agency, it's on the sponsor to come forward and discuss what kind of cross-reactivity there is for the CAR, as well as cytokines that might either be co-administered with the cells or administered just as part of the model. I kind of see that skipped over a lot as clients rush towards a pre-IND where they're not considering as much this cross-reactivity. But again, as it gets harder and harder, then the solutions to those problems sometimes are more expensive animal models that include transgenic mice that overexpress a target antigen, or other models that have to be developed or engineered more specifically. And so I think that's still a major challenge, and it's only getting harder, I think, as some of these products get more complex.
Yeah. Robert, did you have any comments? I know that was a non-clinical question, but did you have any thoughts or anything to add there?
Yeah, sure. I can agree with Chris a lot in regards to the complexity of what's coming through, today versus what we've seen over the past several years. There's a lot of technologies being stacked upon one another, kind of being put into these, and so exactly how one goes about demonstrating that, the non-clinical aspect's its own, but there's still some expectations from the CMC perspective to kind of define those new materials and show that they're suitable for what their design was intended for. And so I think that level of complexity is just an understatement, because there's so much kind of coming through and different vectors being used even on a cell therapy, which ultimately having a ex vivo product is its own complication, and this just adds to it with all the other capabilities that these tools are now being able to provide for these therapeutic events.
Yeah, they're only getting more complicated, and certainly you have the targeting moiety, the signaling moiety, the vector, the cells. So there's a lot to think about. One of the key issues I've heard in the past with other sponsors pertains to vector copy number. I think it's a big deal for FDA. What are some of the key concerns you've seen with vector copy number and how sponsors should best express it? Robert?
Yeah, thanks. Great question. I've seen the evolution of the vector copy number assay over my duration in human gene therapy. And I think that what has become much more the industry trend, and now the guidances kind of give a lot more clarity in, is being able to quantify the copy numbers on a per transduced cell. And so it's a complication again in that regards, because it requires another assay to kind of determine which cells are positive and which cells are negative. And the technology being used, it could be a lack of expression that's being used, or positive expression with a transgene being delivered. And so that marks the total number of cells that are transduced. And then having a PCR-based approach for quantifying the copy numbers, which is pretty common, that allows for the actual copies of a transduced cell to be considered in the studies. And a lot of these developments, again, like we were talking about previously, there's so many complications. You even see multiple vectors coming in, or multiple transducing vectors. And so that kind of pushes the limits towards the total number of copies one can have, and what constitutes a positive cell for one transgene versus another. And then there's heterogeneity in that cell population. And even speaking towards the cell, that's its own thing, because if it's considered a tissue in regards to what's being collected, or if there is like an iPS cell or something that is more of a cell line, it just allows for a lot more control of the manufacturing process. Of course, the more homogeneous the population is, but I think ultimately the copy number just kind of provides a concern for safety. And typically there's a general concern around the oncogenic nature of the vectors, some of them more than others. And with the lentiviral vector specifically, there's been a general cap for an allowance of 5 copies per cell, but that's an average over a population of cells, and so some cells could have more and some cells could have less, and so it's very difficult to kind of attribute exactly what's going on. One thing I don't see too much of is the total number of copies, in a dose and what that confers in that regard. And sometimes I've seen therapeutics that do have some sort of correlation to that metric, because it is about protein expression and not per cell basis. And so, in a long-winded answer, there's a safety concern. Nothing really has emerged of significance, let's say, but ultimately, I think the guidances are giving good ideas towards how to give everybody a fair understanding of what the expectations are.
Yeah, yeah, and Chris, is there any implications of a high or an excessively high vector copy number on non-clinical safety, or does that possibly impact non-clinical safety? And are there any assays that sponsors need to think about?
I think it comes down more to making sure that your material for your pivotal safety studies is representative, and this is an issue that we're dealing with a lot, and it's one where Robert and I overlap with sponsors quite a bit, because if the vector copy number is much higher than in your product, then there's certainly going to be than the material used in your tox study, then there could be an increased perceived risk there. And I think this also sort of ties in with another common issue that we see across, is just how to demonstrate comparability. We now CAR Ts are a global type of product. There are people, sponsors who are doing trials in other countries. They want to come to do trials in the U.S. and there's a change in manufacturing site there, and that's probably the bigger hurdle, is maybe not one characteristic or another around copy number, but how we can really demonstrate that if the safety assessment was done for a product that's manufactured in Asia or somewhere else for another clinical trial outside the U.S., does or does not warrant an additional safety study for farm tox, or a non-clinical safety study to support the U.S. trial.
Yeah, that's interesting. There's so many considerations. And to follow on that, I know that this has been a big issue too. Most of all the products, I think, have been developed for hematologic cancers. What do you see as Chris? What do you see as some of the issues or barriers to developing CAR Ts for solid tumors, and where that sort of is right now? Can you comment on that?
Yeah, I mean, I think it's much easier to manipulate the system for research purposes around solid versus liquid tumors in these mouse models, because you can develop mouse models where you can pretty much do intratumoral injection, or they can have tumors that are easy to access for direct treatment. There are some clinical trials that also do intratumoral injection to get better access for drugs to the tumor tissue. But I think most CAR T products, again, we're still talking about an intravenous administration, and it really comes down to how those cells can permeate into areas of solid tumors and actually drive that immune response. And sponsors are coming up with a lot of different ways to try to increase activity and homing, but at the end of the day, it was sort of low-hanging fruit with the first few products as an intravenous administration of T cells to a compartment that has a blood cancer, was made perfect sense. And now the dissemination of those cells to areas of solid tumors has been a challenge. I'm not sure we really solved it there. Intratumoral injection is one way, but again, that's not something that's easy to model in very small mice.
Yeah, that's definitely a challenge. And I know one of the other challenges has been, developing, all these products have generally been autologous, and maybe, I know there's been a lot of work into trying to develop, allogeneic CAR T products. And I want to leave a little time for questions here at the end. But Robert, what do you see are some of the challenges involved in developing an allogeneic CAR T product versus autologous?
Thank you, Dave. I think that ultimately the allogeneic material on the surface, it would seem like it'd be a bit more straightforward because you're generating a stock material that can be used for multiple doses. But behind the scenes, there's a lot of complications. There's a lot of development that's needed to kind of get to that state where you do have a very, promising therapeutic. And I think ultimately with the autologous cells, the idea that the patients receiving the cells kind of limits the expected testing, but with the allogeneic material and how they're sourced, the guidances have now come with a much more clear understanding in regard towards what the tests are. And so, of course, like adventitious agents and other aspects like the alloreactive lymphocytes, or and kind of even going back to the aberrant growth, that's, they brought that back up. And it is a key feature because these aren't the donor's cells that they're receiving, right? They're not their own cells that they're receiving. They're receiving a donor's cells in that regard. And so being able to kind of understand what the aberrant growth is with these gene modifications, does require some level of genotoxicity at times, and being able to go through even potentially like in vitro immortalization assays or other aspects. Again, this kind of is the gray area between non-clinical and CMC, where there are some checkpoints along the way that you want to, check off as being safe and representative of the material that's going to be in the clinic is the key, because that aspect is the important part that allows for the validation to occur more seamlessly. And I think that those additional tests, are the key differences. But just from a personal experience, there's seems to be a lot more development upstream for the product than there is like with an autologous product. Not that there isn't a lot with autologous products, just that there is a lot overall for allogeneic.
One of the key concerns I've seen is they always, and the FDA cited several times, is graft versus host disease, GVHD. So I think that is one we'll have to keep an eye out for, and it's just something you always have to deal with allo products in general and human cells, right? So, but we have a few minutes left. I don't know if we want to go to questions, or if the audience has questions for our panelists. We can go to those now. Let's see. I don't know if they're written or if you can chime in and be unmuted. Are there any other questions from the audience?
David,
Okay.
David, this is Chris. I don't think there are any
Yep.
questions right now. So why don't we go on with another question for Chris and Robert, if you have another one there.
Yeah. Well, certainly. But so, Chris, I know there's still, just kind of, this is kind of a general question, but what do you see? You probably touched on this a little bit already, but some of the more pressing challenges for non-clinical safety testing of these products. There's a lot of barriers, and the animal models are an issue, but what are some of the other challenges that you've seen sponsors run into?
Yeah, I think beyond, finding the right animal model, probably being the biggest one, is there just seems to be a lack of material for a lot of sponsors who are on a rapid development path. And so lining up your tox studies these days, it becomes actually a logistical challenge. There are, scheduling challenges with finding a CRO to do your tox study, and then you'd want your most representative material available to treat, a few dozen mice with, which is not a small amount of material. Sponsors are often faced with the question of whether to use a less representative material from a research grade batch to try to get safety data earlier, or wait until they have something from like an engineering run or something that's from their actual manufacturer that's going to make their clinical product, which I think would be probably preferred. I'll defer to Robert, but it just has become a development challenge as well, because even if you have the right model, getting material that you can really demonstrate is representative of your clinical product in time for a tox study, which oftentimes takes quite a while to get your final report back, has become, I think, an increasing problem since the pandemic. There seems to just be a lot more pressure on non-clinical CROs to be available, and pressure on finding these contract manufacturers as well.
Yeah. And what about too, if you're using a tumor model, tumor-bearing animal model in immunocompromised animals, you have to, keep in mind the potential toxicities associated with the tumor and the adverse findings, because these aren't normal healthy animals, right?
Yeah, that's a great point. I mean, then you get into challenges with finding a GLP compliant CRO. If you're only collecting safety data in a model that also requires administration of tumor cells, there are fewer CROs who are comfortable. Either they specialize in doing the pharmacology part where you can test efficacy, or they specialize in doing the safety part where there's not usually tumor bearing mice. When you mix the two, it becomes a challenge just to get the study designed properly so the animals survive the study, but also finding a CRO to do that in a GLP setting is even harder. So lots of things sort of coming together at the end there.
Okay. And maybe one final question for Robert, if you don't have any additional questions from the audience, but there's a lot of elements obviously involved in a CAR T product, but how can the actual cell type or transducing vector or the linker, the targeting moiety, any of these elements in your mind impact CAR T function and safety? It's kind of a catch-all question, but
yeah, it is. Yeah, no, thank you. I think that there's a lot that can impact overall when it comes to, let's say, the vector, and like we were talking, the cell type or tissue target that it's being explored, and the nature of like the pseudotyping is advancing quite rapidly, and so there's more cell-specific targeting to that end. Though obviously they're targeting organs and such if it's a direct injection, or even other approaches, as far as like the alternative bearing. And I've seen a lot of research over my 20 years in the pseudotyping for lentiviral vectors, and their capabilities are advancing. And the analytics nowadays are certainly capable of being able to give a good understanding of the different cells that are being transduced and how it impacts them. But ultimately, what has kind of become more popular is the deeper characterizations that can occur because of the whole sequencings of RNAs or genomes and such, and being able to kind of attribute what's occurring there. And so I think that the overall function can be a bit more precise. The safety can be a bit more safe, to kind of keep it at that level. And I think that the target tissues, really do make the bigger difference in regards towards how the therapeutic's going to work. Because when trying to demonstrate these in vitro or in the labs setting, it's difficult to recapitulate exactly what's going to be happening in vivo. And so I think being able to kind of get some clear definition of your vector's potential and showing its selectivity of different targets and such in that setting, it's really providing a lot of benefit towards the whole community in gene therapy, being able to kind of explore different diseases and indications, and it's safe and therapeutic way.
Yep. Chris, did you have any final thoughts? We got maybe 1 minute left if we don't have any questions, so we can cap it off here.
Yeah, I wasn't sure if you were referring to me or Chris Crahan.
Chris,
but
you.
I think we pretty much covered it. I just did want to hit on some of those overlaps between, getting the right material and checking comparability, as well as planning out your farm tox studies. So I think that's important to consider, and it falls in line with the recent guidance from this year.
Well, thank you very much, both of you. It's been insightful, and I'll turn it back over to Chris Crahansel.
Great. Thanks, David. And thank you also, Robert and Chris, for joining us and giving us some great insight. And again, we're just kind of scratching the surface here for a lot of the clients that we work with and sponsors that may want to further assistance from us. And so I just want to say thanks for all those who have joined and those who will also be listening to this recording afterwards. If you have any other questions, or if you need some regulatory support, we are here. Biologics Consulting has been around for 30 years, providing regulatory and product development services across non-clinical, CMC, medical device, regulatory affairs, and e-publishing services. And so we are here at your disposal, and we're here to help and support. And we do all of this for the good of public health. So once again, thank you everyone for joining us. And again, thank you, David, Robert, and Chris. We appreciate your insight and your help here today. Hope you all have a great day, and thanks again for tuning in. Take care. Bye-bye.
Thank you.
Thanks.
You bet.
Thank you.
Bye.